from a vision, of where the single cell genomics field is going. Where does your view on what the real kind of critical next advances in single cell genomics is?
For sure, being able to do a multi-omic RNA and surface protein, intracellular proteins, because in the end we can look at the RNA, but the real effectors are the proteins and chromatin. This, of course, with the higher throughput, ideally. So plate-based methods are still an edge because they have a higher sensitivity, but they have a relatively low throughput. But it depends on the question. But I would say I would like to have a method that is sampling RNA, protein, and chromatin and methylation
Sure.
at a high throughput, lower cost, fast.
Yeah, I think that is certainly top of the list for many people.